Good morning, everyone. Welcome to today's webinar hosted by ECD on how to visualize your research breakthroughs with RNAscope Multiplex Fluorescent Assays. I'm Tapan Shah, a technical support scientist at ACD Bio. Before I begin, I'm going to give you an overview of the topics that I'll be covering in today's webinar. I'll first go over the RNAscope technology overview. I'll then give you a snapshot of different assays. I'll then go on to the multiplex fluorescent assay, which is going to be the focus of today's webinar, and a compare and contrast table on choosing the right fluorescent assay for your needs. I will then describe the RNAscope assay workflow, followed by key guidelines and tips for success with RNAscope assays. And then in the later half of our webinar, I will focus on troubleshooting tips for multiplex fluorescent assays and some examples of successful staining from peer-reviewed publications. And I'll then end it with how to understand your RNAscope data and with some frequently asked questions. Okay, so let's get started. So RNAscope is an in situ hybridization assay, which is a specific and sensitive method to detect RNA in cells and tissues in a morphological context. As I mentioned, it's a hybridization-based technique, and it has simultaneous signal amplification and background suppression system. It allows detection of multiple RNA markers using fluorescent or chromogenic labeling, and it is compatible with a variety of sample types, including FFPE, fresh frozen, fixed frozen, and cultured cells. The key component of the RNAscope technology is the double-Z probe. As shown in figure here, the double-Z probe has a spacer and an upper region. The lower region consists of 18 to 25 base sequence. So the double-Z pair would constitute a 50 base region, which is going to be the target specific binding site. The upper region is separated from the lower region by a spacer sequence. The upper region consists of a 14 base TIL sequence, which adds up to 28 bases for the pair and serves as a pre-amplifier binding site, which is for 28 bases. An ideal target region for an RNAscope probe would be 1,000 bases in length, which would constitute of 20 pairs of double-Z pairs or 20 double-Z pairs, which would be 50 bases each. We will now look at the signal amplification technology that is associated with the RNAscope assays. So as I showed in the previous slide, the lower part of the double-Z probe binds to the target specific region and has a linker that separates the upper part of the double-Z probe, which serves as the pre-amplifier binding site. As I just mentioned, the pre-amplifier would bind the upper region of the double-Z probe, followed by amplifiers binding to this pre-amplifier, which is going to generate an amplification tree. And finally, you can have a labeled probe, which would be an enzyme or a fluorophore to detect the signal. This signal amplification is what contributes to the sensitivity of the assay. Not is the assay sensitive, but it's also specific. By that, what I mean is, if only a single Z is to bind, you will not detect a signal. Or in other words, binding of the double-Z pair is essential to detect a signal. To date, we have more than 1,700 peer-reviewed publications in a variety of peer-reviewed journals, showing the wide adoption of the technology across the scientific community. I will now go through a snapshot of the different RNAscope assays that ACD provides. RNAscope assays can be primarily categorized in two major categories: chromogenic, which is for the first three columns on the table here, or fluorescent, which is the final two columns on the table here. The chromogenic assay can be singleplex brown, singleplex red, or duplex red and green. The chromogenic assays have the capacity to detect only 2 targets or one or 2 targets at a time. Whereas the fluorescent assay, which have 2 versions, version 1 and version 2, can detect up to 3 or even 4 targets respectively. Both chromogenic and multiplex fluorescent assays are available for manual as well as automated platforms such as Leica Bond RX and Ventana Discovery. And as I mentioned earlier, the focus of today's webinar is going to be on the multiplex fluorescent assays, the version 1 and the version 2. I will now give a brief overview of the technology for the version 1 and the version 2 fluorescent assay. The multiplex fluorescent version 1 assay consists of an assay which can detect up to 3 targets at a time and has directly conjugated fluorophores as shown here. Please note C1, C2, and C3 in this case represent 3 different target probes. The multiplex fluorescent V2 assay, which I will be calling on as the V2 assay from here on, has Perkin-Elmer's TSA-based amplification technology and has the capacity to detect 4 targets in a sample type at the same time. Please note, opal dyes are required for the 4-plex capability. We will now go to an important question, which is, which of the 2 RNAscope multiplex fluorescent assay is right for you? Shown on this slide is a compare and contrast table which compares version 1 and version 2 of the multiplex fluorescent assay. The version 1 of the assay is compatible with fresh frozen, fixed frozen, and cultured cells. It can detect up to 3 targets. At this point of time, it is not recommended to be used for IHC and is recommended for low autofluorescing tissues. The workflow length for the V1 assay is 6 hours. As opposed to the V1 assay, the V2 assay is also compatible with FFPE samples, apart from being compatible with fixed frozen, fresh frozen, and cultured cells. The V2 assay can detect up to 4 RNA targets simultaneously. Please note, as I mentioned earlier, opal dyes are required for the 4-plex capability. The V2 version is compatible with the IHC co-detection and is recommended for tissues which have high autofluorescence. As compared to the V1 assay, the V2 assay has a longer workflow length, which is 14 hours, and is usually better off if split between 2 days. I will now go over the assay workflow. All RNAscope assays start with a permeabilization step, which includes the pretreatment to unmask the target RNA in tissue and cells. This is then followed by hybridization of target-specific double-Z probes, which target the RNA molecules. And then you will have a sequential hybridization of amplifiers and labeled probes, which constitutes the amplification step. And you'll finally visualize your RNA molecules in a tissue context using microscopy. And you can quantify the RNA molecules which are observed as punctate dots. We will now look at the assay workflow for the V1 and the V2 assay. We'll start with the V1 assay. Please note the workflow here is shown for fresh frozen tissues. The V1 assay, just like all other RNAscope assays, starts with a pre-treatment step, which includes a post-fix and a protease incubation. This is then followed by hybridization of the multiplex probe, and you will then have the amplification steps, which consist of AMP1, AMP2, and AMP3. Finally, you will label the probe using AMP4 Alt A, B, or C. Please note, for the V1 assay, you have to choose only one of the AMP4 Alt options, meaning you will choose either AMP4 Alt A or B or C. Following the AMP4 Alt incubation, you will stain the nuclei with DAP-B, mount with prolonged gold, and cover slip. Shown here are the three different configurations for the fluorophores possible based off your AMP4 Alt selection, which would be AMP Alt A, AMP Alt B, and AMP4 Alt C. To make things simpler, as you can see, Alexa 48E8 is in three different Alt combinations. Is in channel 1 for Alt A, channel 2 for Alt B, and channel C for Alt C. Here is an example of a successful 3-plex staining using the V1 assay for human HeLa cell line. Shown here are three different housekeeping genes: POLA2A, PPIB, and UBC. We will now move on to the assay workflow for the V2 assay. Please note, the workflow shown here is for FFPE tissues. The V2 assay would start with the pre-treatment step, which would consist of baking, deparaffinization, hydrogen peroxide treatment, target retrieval, followed by protease plus. After the pre-treatment, you will hybridize your multiplex probe and then do the amplification steps, which include AMP1, AMP2, and AMP3. You will then move on to develop the fluorophores, starting with the first fluorophore by incubating with HRPC1, followed by incubation with TSA plus fluorophore, followed by HRP blocker, and you will repeat the three steps for the second fluorophore and the third fluorophore. Please note, with the V2 assay, you have the option of mixing the fluorophores to be associated with the channels, meaning you can have site 3 or site 5 for HRPC1. Since the assay work length is longer for the V2 assay, it's advisable that it is split over 2 days. An ideal stopping point would be after you hybridize your probes. Post probe hybridization, you can store your slides in 5x sodium saline citrate buffer overnight at room temperature and begin the amplification steps the following day. Please note, irrespective of the number of targets that you're trying to probe for, all AMP3 steps or AMP1, AMP2, and AMP3 steps must be completed. Failure to do so will result in weak or no signal. The multiplex V2 assay can be used as singleplex, duplex, or multiplex. Meaning, if you were to develop only one target, then you will do HRPC1, incubate with the TSA plus fluorophore of your interest, do the HRB blocker step, and then proceed directly to DAP-B to counterstain the nucleus, mount with prolonged gold, and cover slip. Shown here are the fluorophores that you will need to buy separately from PerkinElmer for the V2 assay. On the left is shown the TSA plus fluorophores. On the right is shown the opal dyes. Please note, for the 4-plex capability, you will need to use opal dyes. The recommended dilution range for both TSA plus fluorophores and opal dyes is 1:750 to 1:3000. For the 4-plex assay, we recommend using multispectral imaging systems, including Parkine Hermos, NEURONS, Mantra, or Vectra systems. On this slide is shown a successful 4-plex staining with the V2 assay. The sample type shown here is FFPE sample from a liver cancer, and as you can see, 4 different genes are shown in 4 different colors: green, yellow, red, and white respectively. I will now talk about things that you need to get started with your RNAscope assay. Here are some key guidelines for RNAscope assays. We strongly recommend that you follow the protocols exactly as described in the user manuals. Please review the sample pretreatment conditions and ensure that you use the right sample pretreatment condition for your sample type. We strongly recommend that you always use control probes, positive and negative, to qualify the RNA and the sample quality, and review the checklist to ensure that you're using all the required materials. You can visit the link shown at the bottom of the slide for tips on getting started. Shown on this slide are the pictures for different reagent kits from ACD. On the far left, you have a picture of pretreatment reagent kit. In the center is the multiplex fluorescent detection kit, and on the far right is the wash buffer kit. Please note, if you see precipitation with the 50x wash buffer, incubate it at 40 degrees Celsius for 20 minutes to dissolve the precipitate. For the 4-plex multiplex fluorescent V2 assay, please use the RNAscope 4-plex ancillary kit. On this slide is shown how can you qualify your samples using controls. We strongly recommend that you use control slides and control probes to verify your technique and do the sample and RNA quality check. On the left is shown control slides for HeLa cell pellet. On the top is shown a picture for the negative control, where it only shows the blue staining for the nucleus and no RNA staining or no puncted dots. And at the bottom, you see the multiplex positive control probe showing the RNA as puncted dots, green and red. Once you have verified your technique, you can then proceed to use control probes and qualify or quality check your sample and RNA. In this case, for the tissue sample, we are looking at fresh frozen human brain. Again, for the negative control, you're seeing only the nucleus stain, and for the positive control, you can see RNA staining as puncted dot. Once the technique and the sample or RNA quality check has passed, you can run your target probes. If it fails, we recommend that you first verify the technique, check RNA quality using new samples, and perform assay optimization if necessary. Shown here is an example of a 3-plex positive control staining to check the RNA quality. The image is shown for fresh frozen mouse brain with the V1 assay. 3 different housekeeping genes are shown for 3 different colors: POLR2A, PPRB, and UBC. Please note, the negative control is equally important as the positive control. The negative control essentially serves as blank, and would be required for background subtraction when doing the quantification. The table here shows the details for the RNAscope probes. The 3-plex positive control probe consists of 3 different housekeeping genes: Polar 2A in channel 1, PPIB in channel 2, and UBC in channel 3. The negative control probe is for the DAB-B gene, which is isolated from soil bacteria. And then you have the target probes, which are specific for the gene. You can visit the link shown in the 3rd column for RNAscope catalog probes and made-to-order custom probes. Please note, all channel 1 probes are provided in ready-to-use dropper bottles and require no dilution. However, channel 2, channel 3, and channel 4 probes are provided in a 50x concentration and have to be diluted using the C1 probe. If no C1 probe is used, please use the probe diluent to dilute C3, C2, and C4 probes. Please ensure that you warm the probes at 40 degrees Celsius for 10 minutes before use. This slide, as I shown previously, shows the TSA Plus fluorophores and the opal dyes that have to be purchased separately from Perkin-Elmer to be used with the V2 assay. We will now review the checklist for doing the RNAscope assay. Some of the key points to note on this checklist are: always use superfrost plus slides, always use fresh reagents, use the high-BZ oven and accessories, always use positive and negative controls to qualify the RNA quality in your sample, and always use mounting media, which is prolonged gold, for fluorescent assays. Shown on this slide are the recommended accessories for the RNAscope assay. On the top panel are shown the accessories that are available from ACD. You have the Easy Batch system for slide processing, the high-BZ oven, which controls temperature and humidity and is essential to the success of RNAscope assay. On the bottom panel, you have tissue track washing tray and then hot plate or steamer for target retrieval. We will now look at some of the workflow guidelines that should be followed. A couple of important guidelines from the list include: please perform all the M steps in the right order and do not skip them. Failure to do so will result in weak or no signal. Do not let the slides dry out unless specifically mentioned in the protocol. We will now go through some troubleshooting tips for the multiplex fluorescent assays. Shown on this slide is a successful 3-plex staining with the V1 assay for lung cancer tissue sample. The V1 assay is recommended for fresh frozen samples. As I mentioned previously, the C1 probes are in 1x concentration by default, while C2 and C3 are 50x. To make a 3-plex probe mixture at 1x concentration, please dilute the C2 and C3 probes 50 times using C1 probe. If using only C2 and or C3 probe, use the probe diluent and mix at a 1:50 dilution. Please note, we recommend prolonged gold as the mounting media for the fluorescent assays. Shown here is an example of successful 4-plex staining with the V2 assay. The sample type shown here is FFPE sample from liver cancer. The V2 assay is recommended for FFPE and low expressing targets, primarily because it has Perkin-Elmer's TSA-based amplification technology. With the V2 assay, the customers have an advantage to detect up to 4 mRNA markers in a single tissue section. As I mentioned previously, we recommend Prolonged Gold as the mounting medium. Please note, the Fluorophores will have to be bought separately from Perkin-Elmer for use with the V2 assay. One of the common troubleshooting parameters with the fluorescent assays is the issue of autofluorescence. Several tissues tend to have inherent autofluorescence, shown in the left here, such as mouse kidney, intestine, colon, and brain. The tips for this would be using V2 assay. Adjust the TSA concentration starting with 1:1500. Please assign the highest expressing target in the green channel for better signal to noise ratio, and use multispectral imaging software to subtract background autofluorescence. The other troubleshooting parameter is sample digestion. The tips to resolve this is use the recommended protease condition as per your sample type. Please note, overdigestion can cause autofluorescence, which may mask your staining, which is evident from the upper left panel showing fresh frozen mouse kidney. However, when optimal protease and pretreatment conditions were used, you can now see a better signal to noise ratio, and you can now visualize the RNA molecules as puncted dots shown in upper right panel. And similar is the case for the lower panel showing fresh frozen mouse brain sample. We will now look at the troubleshooting tips for sample detachment. We will be reviewing the sample detachment troubleshooting tips based on the sample type. On this slide is shown the sample detachment troubleshooting tips for FFPE samples. If you see tissue detachment with FFPE samples, we recommend that you do an additional baking step for 30 minutes at 60 degrees Celsius after the deparaffinization. If required, you can do an additional baking step again for 30 minutes at 60 degrees Celsius after you perform target retrieval. Please note that we recommend using Superfrost Plus slides. Please bake or dry the slides in a circulating air oven and maintain a boiling temperature of close to 100 degrees Celsius during the target retrieval. For fixed frozen samples, we recommend that you air drying the samples at -20 degrees Celsius for 2 hours instead of 20 minutes in case you're seeing sample detachment. Additionally, you can bake the slides at 60 degrees Celsius for 1 hour after you air dry them. You can also try a post-fix in 10% NBF for 1 hour at room temperature after the target retrieval step. If none of these 3 modifications work, you can omit the target retrieval and extend the protease time to 45 minutes. Please note the recommended section thickness for fixed frozen samples is 7 to 15 microns, and again use an air circulating oven to bake or dry slides, and ensure that the temperature of 100 degrees Celsius is maintained during the target retrieval step. For fresh frozen samples, if you're seeing sample detachment, we recommend that you extend the fixation time to 1 hour to 1 hour 30 minutes at 4C instead of 15 minutes at 4C. Additionally, you can try a baking step for 30 minutes at 60 degrees Celsius after you dehydrate your sections. We'll now look at some of the common multiplex fluorescent issues. One of the most common issues is no or weak signal. There could be 3 contributing factors: microscope, the assay, or the sample. With the microscope, the problem could be that you're using the wrong filter, wrong exposure time, or inappropriate image enhancing with the software. The solution is use the correct filter settings. Do not use auto exposure at first. Verify the signal with naked eye and use multispectral image enhancing software, for example, NURONS. With the assay, the problem could be that you're missing the M steps. The solution is to ensure that you complete all 3 M steps, and this is important regardless of the number of targets that you're probing for. The third source for no or weak signal could be the sample. It could be the RNA quality in the sample is compromised or the sample preparation is not correct. The solution would be use a new sample with good RNA quality, follow the pretreatment guideline as recommended, always perform the positive and negative control probes to assess the RNA quality, and always check the signal first with naked eye under the objective lens. We will now look at some examples of successful staining from peer-reviewed publications. Shown on this slide is a study from Satyamurthy et al. Published earlier this year in Cell Reports, where the authors were looking at single nucleus transcriptional profiling in spinal cord neurons. Using single nucleus RNA-seq, the authors identified 43 different neuronal populations, as shown in the figure on the left. The authors also identified novel genes in some of these neuronal populations, and these genes, the novel genes, were confirmed using RNAscope assay. So shown on the right is the multiplex fluorescent RNAscope assay. In the DE5 cluster, which is highlighted in blue on the left image, the authors were able to validate the expression of 2 novel genes found in that cluster, namely SNCA and NECAB1 and CACNA1E and SYT1 for the DL1 cluster shown in red. Therefore, the take-home message being the RNAscope assay was used for the validation of co-labeling for pair of genes that belonged to the specific neuronal cluster. In this study from Branco et al., published in Cell in 2016, the authors were looking at the expression of sodium gated channel in AGRP and POMC neurons and how that regulates body weight. Using the multiplex RNAscope assay, the authors were able to show that SCN9A, which is a marker for sodium gated channel, showing that the SCN9 is expressed in both AGRP and POMC neurons. The AGRP is shown in the top panel and POMC is shown in the bottom panel. In this study, published by Herring et al. In Nature Neuroscience earlier this year, the author focused on molecular and spatial analysis of sensory neurons in the dorsal horn. Using single-cell RNA-seq, the authors identified 15 different molecular subtypes of the glutamatergic neurons. The focus here is on 3 molecular subtypes, GLUT5, 6, and 7. Using RNA-seq, they were able to confirm that the GLUT5 subtype and the GLUT6 subtype had high expression of TAC2, whereas the GLUT7 subtype had a low expression of TAC2, which was confirmed using RNAscope as shown in panel B, the right image, column 2. As you can see, for TAC2 staining in green, the expression is high for GLUT5 and GLUT6, but it's low for GLUT7. The authors were able to confirm the staining for NMU gene as well. The single-cell RNA-seq showed that GLUT5 subtype was negative for NMU, and GLUT6 and GLUT7 were positive for NMU, which is validated, as I said, using RNAscope assay shown in column 3. SST here is the neuronal marker. Therefore, the take-home message being RNAscope multiplex assay, multiplex fluorescent assay was used to validate RNA-seq results in vivo. One of the major advantages of the multiplex fluorescent assay is its capacity to be combined with IHC workflow, or the fact that you can do dual RNAscope-ish IHC workflow. Please note that at this point of time, we recommend using the V2 assay for ish IHC. All dual ish IHC protocols would require optimization. We advise that you perform the ish first followed by IHC, and that you optimize IHC separately using RNAscope pretreatment reagents. Please note the dual ish IHC would work better for highly expressed proteins due to protein degradation during the RNAscope pretreatment. I will now show you a couple of examples of peer-reviewed publications where dual-ish IHC has been used. In this publication from Herring et al. In Nature Neuroscience, the authors used dual RNAscope-ish IHC to look at FDZ10 mRNA expression in developing central nervous system, and they did IHC to look at GLUT1 protein, which is a blood-brain barrier marker. The authors looked at mid-gestation embryos, which were day 12.5 and 14.5 old. The FDZ10 mRNA here is shown in red, and GLUT1 protein is shown, I beg your pardon. The FDZ mRNA is shown in green, and then the GLUT1 protein is shown in red. In another study published by Johnson et al. In 2016, where authors focused on SCHWANN cell precursors and the factors that were secreted from the cells and required for digit tip regeneration. Using ish IHC, the authors were able to show that OSMR mRNA is expressed in PDGFR positive cells, shown on the bottom right. The OSMR RNA is shown in red, and PDGFR protein is shown in green. Moreover, the authors were also able to use RNAscope to support their finding, which showed expression of the ligands increased following amputation of the digit tip, and it then decreased when the regeneration was complete, therefore helping the overall take-home message of the paper. We will now look at some guideline for fluorescent quantification. Some of the key questions for assay and signal are: what does a dot mean? What is the significance of dot size? And what is the difference between a dot and a cluster? With the RNAscope assay, the mRNA molecules are detected as single punctate dots shown in the figure on the left. Each dot represents a single copy of mRNA. Clusters can result from overlapping signals, which can come from multiple mRNA molecules. Please note that there can be variation in dot size or intensity based off the number of ZZ probes that are bound to the target molecule. We will now look at how to quantify the RNAscope assay. A semi-quantitative scoring guideline can be used for quantification of the RNAscope assay, wherein you can give a score from 0 to 4, where 0 would be no staining or less than 1 dot per 10 cells, and then sequentially increasing to a score of 4, which would be more than 15 dots per cell, and greater than 10% of the dots are in clusters. For more information on data analysis, please visit our hyperlink shown at the bottom of the slide to get the data analysis guide. Alternatively, you can also use the Halo software to quantify your signal. The Halo software allows for automated dot, cluster, cell detection, and supports the 4-plex fluorescent ISH detection. For more information on Halo software, please contact Indica Labs. Here are some resources that would serve as a guideline for quantifying the RNAscope fluorescent staining. You can download the guideline on how to quantify RNAscope fluorescent assay results using the link shown here. And you can also download our data analysis guide. And you can watch the webinar entitled Get the Most Out of Your RNAscope Multiplex Fluorescent Assays as well, following the link shown here. And as I mentioned previously, please contact Indica Labs for more information on Halo software. We will now go through some frequently asked questions for the multiplex fluorescent assay. Which amps should I use to develop each channel? Please use all the amps provided in the dissection kit. Each amp reagent serves as a building block for the amplification tree, and if any amp is left out, this may result in no signal. For fluorescent multiplex assay or the V1 assay, please choose only one AMP4 alt configuration depending on your fluorophore preference. For the multiplex fluorescent V2 and the Alice multiplex fluorescent assay on the automated Leica platform, please use all materials provided in the kit. I am seeing bleed through between the fluorophore or channels in multiplex fluorescent V2 assay. How can I avoid this? Use optimized fluorescence filters to minimize fluorescent bleed through. Reduce THA plus fluorophore concentration of the channel that is bleeding through. Reduce the exposure time during image acquisition to avoid overexposure. What to do if RNAscope signal cannot be distinguished from autofluorescence using fluorescent multiplex assay or the V1 assay? If the signal is weak and masked by autofluorescence, please put the highest expressor in Alexa 488 channel. We recommend that you use multispectral imaging systems such as Nuance, Mantra, or Vectra to subtract background autofluorescence. Can I run multiplex fluorescent assay with C2 and C3 channel probe only without C1 probe? Yes, it is possible to do so. However, please note, since C2 and C3 probes are provided in 50x concentration, you will have to dilute them 50 times using the probe diluent. If I'm using FFPE samples, which fluorescent assay is right for me? For FFPE samples, we recommend using multiplex fluorescent V2 assay or the ALX multiplex fluorescent assay. This is because the V2 assay utilizes TSA-based amplification and provides a high signal-to-noise ratio. Can I perform IHC using RNAscope multiplex assays? Yes, it is feasible. However, please note, we recommend that you run the IHC using multiplex fluorescent V2 or the LS multiplex fluorescent assay. Both these assays take advantage of the TSA plus or opal fluorophores, which are designed to be resistant to standard IHC staining protocol and can be viewed for co-expression of mRNA and proteins. Why did my C2 and C3 probes come in a smaller volume? As I mentioned earlier, C2 and C3 channel probes are in 50x concentrated format and should be diluted 1:50 before use. With that, I come towards the end of today's webinar. I strongly recommend that you contact ACD technical support should you have any questions or concerns. You can contact us via email at support.acd@bio-techne.com or via phone from 8 a.m. to 6 p.m. Pacific Standard Time. You can also visit the link shown here for support resources. With that, I would like to thank all of you for listening to today's webinar. We are now open for questions. Please address your questions to all panelists in the chat box, and they will be happy to answer the questions that you have. Once again, thank you for listening. I'll just stop recording, right?